Thank you very much. It's really a great privilege to me to introduce Miklós Sahin-Tóth, our guest from the US, but you may realize immediately that his name is not accidentally Hungarian, because he's a Hungarian origin. He was born in Budapest and went to school at the Semmelweis University. And after that, he moved to the United States and started working on the genetics of pancreatic diseases. And we just met actually with Miklós accidentally in 2003 in a conference in Milano. And after the talk, I went to him and asked whether he would like to be a collaborative member of the University of Szeged and start working in the pancreas together. And he was very keen doing it. So in the last 10 years, he every year many times visited Szeged. He accepted many students in his lab from Hungary. So until now, more than 10 students were working in his lab. And at least, I think, 4 or 5 PhD students graduated in a collaborative projects between the Boston University and the University of Szeged. The other fact is, when you looked at the program, the St. Georgi Mentors programs, you have not only mentors from the Biological Research Center and the Faculty of Medicine, but if you look at, there are many international mentors who are keen to have talented students from Hungary and educating them in his laboratory. So Miklós is one of the international mentors also of the school. And it has to be also stated that he is a really distinguished scientist, has many papers in Nature, Genetics, and other distinguished journals. And I have to say that he is not only, we have not only a scientific cooperation with him, but he's a very good friend of us. And I would like to ask Miklós to start with the first presentation of this event.
Well, thank you, Peter. It's really nice to be here. And when I first got the invitation, I was thinking, what am I doing here? You know, this is the Nobel Prize winners event. But then it really turns out to be, you know, connections to Szeged. And Szeged is almost my second home. I spent so much time here. And so many students came through my laboratory. And really the reason I'm here just to tell you that the laboratory at Boston University is open. And if you want to come, you're most welcome. And all you have to do is write an email to Peter. And he will take it from there. So the next 20 minutes in a sort of very simple set of slides, I will just explain what we really do. And this is a very specific so-called translational research project, which means it focuses on a specific clinical problem. We use basic science methods to figure out how a clinical disease, in this case pancreatitis, occurs. And we try to use this information to help patients eventually. And that's where the name translational comes from, to translate these basic science findings into the clinical application. So the pancreas is the most important organ of in the human body, and the reason you can actually know this because it's right in the middle. If you look at this little guy here, and it's, from either dimension, the pancreas is in the center of the body. And this is not by accident. This really signifies the importance of the organ. Compared to its importance, it's relatively understudied. There's very few groups who study the pancreas. And part of the reason that I come to Szeged so often, that one of the major centers in Europe and in the world which study the pancreas with a long tradition is in Szeged. And this is now led by Peter Hegyi, but before Peter, others have done excellent work. So Hungary is really one of the best pancreatic research centers in the world. So in essence, the pancreas is a big digestive gland which does a very simple job of producing digestive enzymes which normally go to the gut and then help digesting food. Now it's easy to think that all these digestive enzymes can digest also the pancreas. And there's lots of mechanisms to prevent that. One of the most important ones is that the enzymes are made in an inactive form typically, and they get activated in the gut. But if something goes wrong and enzymes get activated inside the pancreas, you get pancreatitis. And that's the inflammatory disorder of the pancreas. And this is a very specific mechanism for the pancreas, since all the other organs do not express digestive enzymes. So you cannot get inflammatory disease in the other organs through self-digestion. So this process, which we call self-digestion or autodigestion, is a very interesting and pancreas-specific mechanism of inflammation. Our interest is mostly how does genetics determine if we get pancreatitis or if we don't. And interesting, different mutations in different genes, which I will show you later, are found in these digestive enzymes which the pancreas produces. So all these mutations, again, very specifically target the pancreas and do not affect other organs. I like to point out just quickly that there's an interesting anatomical arrangement here. So the bile duct, common bile duct, and the pancreatic duct have a common opening. So biliary disease, gallstones is a major cause for inflammatory disorders of the pancreas, which is not a genetic problem, so we don't study that, but this is something Peter Hegyi studies, for instance. Major risk factor for pancreatitis is alcohol, chronic alcoholism, and genetics. So in the clinic, sort of simplistically speaking, there's 3 different kinds of pancreatitis. You can get a single acute episode of pancreatitis, which in an uncomplicated case, it lasts about a week or 2 weeks, and you're most likely you get hospitalized. And it comes with abdominal pain, and there's elevation in the blood of different pancreatic enzymes. Typically, amylase lipase is measured in the clinic. And if there's no complications, then it heals on its own, and then you go home. And the large majority of these cases, they don't get repeated. But there's a fraction of patients who get what's called a recurrent acute pancreatitis, and this is shown here, when you get repeated attacks. And particularly in childhood, this could be very, very unpleasant. It's not life-threatening usually, but the quality of life of people who get recurrent attacks is really poor. At one point, these recurrent attacks damage the pancreas to a point when you start talking about chronic pancreatitis, when you have morphological changes, you have fibrosis, you have ductal irregularities, you have ductal stones, and then you have chronic pancreatitis, which is a risk factor for pancreatic cancer. 20, 30, 40 years after the onset of chronic pancreatitis, there's a very high probability of developing pancreatic cancer. So this is sort of a disease continuum. Interestingly, the genetics determines the recurrent acute and chronic pancreatitis cases. In single episode acute pancreatitis cases, there's no real genetic risk factors, and pancreatic cancer is really determined by other types of genetic changes. So the ones we are interested in is really focusing on this particular disease group. So just to give you some specifics, so these are the proteolytic digestive enzymes which the body, the pancreas produces. And as I mentioned, these are all made in inactive form. So for instance, trypsin is our most abundant digestive enzyme. It's made as trypsinogen. That's the inactive form of trypsin. And chymotrypsins, elastases, carboxypeptidases are all made in inactive forms. So the way that activation can only occur in the gut, this is ensured by a spatial separation of activation by this enzyme enterokinase, which is a specific trypsinogen activator. This enzyme doesn't do anything else, just activates trypsinogen to trypsin. And this is only localized to the brush border membrane of the duodenum. And once trypsin is activated, trypsin is a common activator of all the other digestive proteolytic enzymes. So interestingly, trypsin has the ability to undergo autoactivation, and this little arrow indicates that. So trypsin can also activate trypsinogen. And this mechanism, which is not an efficient mechanism, but this can take place inside the pancreas, and that can lead to pancreatic inflammation. The other point this slide also makes that there's multiple isoforms of each of these enzymes. And isoforms, they almost look like each other, but they are a little different. And from a digestive standpoint, it doesn't matter too much because they all do the same. But from disease pathogenesis, different isoforms do different things. For instance, PRSS1 is one of the major trypsinogen isoforms. This is the only one important for pancreatitis. And among the chymotrypsins, Chymotrypsin C is an important enzyme when it comes to pancreatitis, not so much the other chymotrypsins. So a little bit about history, and then I was trying to give a sort of personal touch to this talk, and so how I got involved in this whole project, and then of course meeting Peter in 2003 was an important aspect of that. But even before that, I was interested in pancreatitis. So if you look at this whole field starts in 1996, that's a big year for pancreatitis genetics. Even though back in the 1950s, people noticed that in some families, pancreatitis is inherited. And then this is called hereditary pancreatitis. There's an autosomal dominant inheritance pattern, almost a Mendelian type disease. And in 1996, they map the gene, but they can only find that it's on chromosome 7. There was no other information at the time. And they got extremely lucky because there was no genomic sequence available at the time, except for this particular locus, which was a T-cell lymphocyte receptor beta locus was sequenced. And the trypsinogen genes are at this particular locus. And nobody understands why, because this is an immunological locus. But this is where trypsinogens are. And that's when they found the first mutation. And this was the article published in 1996. And that's when the field really started developing. And I will show you some more details on that. So the reason I got into this whole thing is actually it's not that exciting that we developed this simple affinity chromatography method. And Ecotin is a protein made by E. coli. And E. coli secretes this protein, and it's a trypsin inhibitor. And it protects E. coli when it's in the gut. And we actually purified this protein and we immobilized it into a column, which is shown here. And what it does, you can actually purify trypsin and trypsinogen in very pure quality and high quantity very easily. And we had this method, which I was working at a company at the time, and they wanted some method, so we did this sort of. It was, and when I read the articles about pancreatitis, I was thinking, well, I got a method to purify trypsinogen, and there's mutations which cause disease. Why don't I just try this? So I started working on this, and I was in somebody else's laboratory, so I only could do this at nights. So I would come in the weekends and stay 3 o'clock, 4 o'clock, late at night, and just purify my little trypsinogen and try to figure out why mutations cause pancreatitis. And this really started out initially back in 1999, 2000 with these single author JBC papers, which was really the product of this late night sort of secretive research on pancreatitis. And initially, I didn't even have a human clone, so the first paper was done on a rat trypsinogen, which turned out behaves completely differently from the human. So that study is irrelevant, actually, we know today. And then later on, we just had an interesting discussion with Eric, about how scientists really exist in a community and how interaction is important. I was so alone with this project that I actually reached out to my father, who was a professor of biochemistry, and we started working together for a few years. So our first important paper, which really I published with my father, who was in Budapest at the biochemistry department, where we really started identifying mechanistic details, what these mutations did. So coming back from this personal aspect to science, so now I'm extending the history of our field. This is pancreas genetics, and I'm not going to go through this slide in detail, but what I'd like to show you that after the initial discovery of trypsinogen mutations, many other different gene mutations were discovered, different risk genes, and every 2, 3, 4 years a new paper comes out with a finding. Now, of course, there's an end to this, and more recently, it's less and less frequent what we find when you find the new genes, but different questions come up instead of just finding new genes. So based on all these genetic studies, the following interesting pattern of mutations emerged. Depending what kind of clinical picture you have, you may have a different set of mutations causing your pancreatitis. And what people found is that if you have inherited almost dominant pancreatitis, then you're likely to have a single mutations with a very big effect. But with other forms of pancreatitis, when you have no family history, we call it idiopathic disease, meaning for unknown reasons, then you're likely to have multiple mutations with smaller effects. And if you go down the list and you reach alcoholic chronic pancreatitis, then you're likely to have a large number of mutations with very small effect, plus alcohol. So now you have an environmental effect, and I can add smoking to this, which is another risk factor. So there's an interesting correlation between the number of mutations you carry and the effect size. Typically, what you would see in the clinic, because dominant cases are very rare, these are the groups you can see in the clinic, and these would have 2, 3 mutations in different genes. A little biochemistry, because this is what what we love to do. So this is the mechanism how trypsinogen activates in a test tube, and this is what we believe happens inside the pancreas. And this is a typical autoactivation curve. If you purify trypsinogen and you leave it at just 37 degrees, within an hour it converts itself to trypsin. So there has to be mechanisms inside the pancreas to stop this from occurring. And one of the mechanisms, which is very important, that we're secreting enzymes, we're flushing it out of the pancreas. So by the time it can activate, then it reaches the gut. There's other mechanisms which we and others discovered. This is the simplest one. This is a trypsin inhibitor made by the pancreas. It's called SPINK1. So if you have a trypsin inhibitor present, then it delays the activation. At one point, the pancreas runs out of inhibitor and the same activation can take place. But as long as you have this, you protect the pancreas. If you have a mutation in this gene and you don't have enough SPINK1, your risk of pancreatitis goes up. There's been a lot of speculation that another mechanism which would be really good to protect the pancreas if you could just get rid of the trypsin, if you just degrade it. And for years, people have been looking for this enzyme. And actually, we did find it in 2008. And this was our paper back then. This turned out to be another digestive enzyme. It's called Chymotrypsin C, which degrades trypsin and trypsinogen with very high specificity. And I'd like to point out this was one of the first collaborative studies with people from Szeged, with Peter and a student who came to Boston. And then this has been an ongoing pattern since then. Also note the number of co-authors on this paper, which is very typical on genetic papers, particularly with rare diseases where you have to have many people contribute samples so you can actually build a strong case about your findings. So what Chymotrypsin C does is really a different mechanism of protection. It doesn't allow activation of trypsinogen, but degrades it, so you get much lower level of trypsin activity. If you have mutations in Chymotrypsin C, you don't have this protective mechanism, you will have pancreatitis much more likely than people who don't have mutations in Chymotrypsin C. So if all these trypsin-dependent mechanisms, trypsin mutations, the trypsin inhibitor mutations, trypsin degradation, Chymotrypsin C, if you put them together in a simple mechanistic form, and I will come back to this just in a second, then you get this picture which says that trypsin is in the center of driving pancreatitis. If you have a mutation in your trypsinogen, then it's easier to get activated to trypsin. And if you have mutations in the protective mechanisms, trypsin inhibition or trypsin degradation, then you get higher trypsin levels and you're higher risk of pancreatitis. To support this model, there's a very interesting mutation in another trypsin isoform. It's called anionic trypsinogen. This is a protective mutation, and the Hungarian population, about 3 to 5% carries this mutation. And if you have this mutation, some of your trypsinogen gets degraded spontaneously. And this spontaneous degradation actually protects you from ever getting pancreatitis. It's not a huge protection, but it is some level of protection. So the slide which I skipped, and I'm going to jump back to it, we were really puzzled that how can digestive enzymes regulate each other? Because these are very, very non-specific. They digest food, so they don't make any distinction between different substrates. Still, we found that Chymotrypsin C very specifically degrades trypsinogen. It doesn't degrade any other enzymes, and other chymotrypsins don't degrade trypsinogen. It's a highly specific interaction. And what we figured out through a number of studies, including crystallization, that there's an attraction between Chymotrypsin C, this is Chymotrypsin C, and this is trypsinogen. And this is really, if you watch Star Trek, this is really like the tractor beam, if you remember that, which from very long distances can attract two enzymes together. And once they come near each other, then the binding is not specific anymore. It just recognizes just like any other substrate. But what gives specificity to this interaction, this initial long-range electrostatic interaction. So this model of trypsin-dependent pathological pathway in pancreatitis is very well established today. What we don't understand really is what does trypsin do? You have this trypsin generated and it digests something, but we're not sure what it is. We still have some theories. So one hypothesis which actually Peter Hegyi developed, and I think we are in agreement on this one, maybe it's the only thing we are in agreement on, right, is that maybe it's a cystic fibrosis-like disease. So cystic fibrosis is an inherited condition when you have mutations in your CFTR, the cystic fibrosis ion channel. And what happens, you get plugs in your duct because there's no flushing, there's no flow in your duct. And this is the picture you get. These are big dilated ducts. It's all plugged up. And by the time you're born, usually you have your pancreas destroyed. And we believe that maybe what happens in hereditary pancreatitis and other forms of pancreatitis, you get protein plugs due to precipitation of some proteins called stone protein or inhibition of this cystic fibrosis ion channel, and you end up with ductal obstruction, and then you had pancreatitis. This is only a model at this point, and we need more evidence that, to prove it, but it's a very, very lucrative sort of working model. The other question which came up over the years is that is trypsin the only mechanism you can get pancreatitis? And it turns out it's not. And this is a more recent paper where we actually identified carboxypeptidase A1, which is another digestive protease, as a major risk factor for pancreatitis. The surprise came when we believed that this is a digestive enzyme, so it will digest something. The effect of carboxypeptidase A mutations is through a completely different mechanism. It's through misfolding. Somehow the protein cannot fold correctly, and what it causes is endoplasmic reticulum stress inside the cell. And ER stress, endoplasmic reticulum stress, increases risk for pancreatitis again. So this is a mechanism independent of trypsin. Again, I'd like to show there's a number of people from Szeged who participated in this study. And what you can also see that compared to 2008, the number of co-authors has doubled. And what it really shows is that now you have to put in a lot more effort to publish a high-level paper. If you want to send in this paper in 2015, you have to add another 50 co-authors, for the same journal, most likely. So to summarize, what I'd like to just discuss and convey today is that risk for pancreatitis is mediated by a number of risk factors which really fall into two kinds of of mechanistic pathways. There's a trypsin-dependent pathway where trypsinogen mutations, that's PRSS1, trypsin inhibitor mutations, pink one, or the trypsin degrading enzyme mutations, these all cause increased trypsin activity and increased risk, maybe through a cystic fibrosis-like condition. There's an alternative pathway which through misfolding and carboxypeptidase A mutations fall here, which cause endoplasmic reticulum stress. And the mechanism is not really clear how to increase your pancreatitis risk. The important thing is now we can use this framework to really go back to patients and offer them genetic screening and genetic counseling based on this. And this is something which Peter started doing also here in Szeged. And I think for about 2 years now, the genetic pancreatitis genetic screening and counseling has been very successful. We also maintain a website. So if you're ever diagnosed with a mutation and you're interested in what it does, you can go to this website, click on a particular risk gene, and under each you will find 50, 60, or 100 different mutations. So we give information about all these mutations. So this is primarily a research tool, but I think also in genetic counseling this comes in handy. Now this is the most important slide, and the final slide is that what is exactly what you need, to be a successful scientist. Well, first of all, I'd like to point at this picture that you must be able to work with some really shady characters sometimes, but it pays off. Second, you need funding, and these are the different grants we got from the National Institute of Health, which is the major funding source in the United States, and we've been very fortunate and blessed that. But most importantly, you need good people, and we get a lot of students from Hungary, and most of them from Szeged, and I put all these names which came through, the laboratory over the past, 12 or 13 years, and the ones highlighted in red, those actually got their PhD degrees in Hungary based on the work they did in Boston. I was their one of their advisors. And these are some of the collaborators here in Szeged who have been, particularly Peter, Zoltán Rakoncai, Mandivet, I've had the fortune of working together with. And I thank you very much for your attention.